Hi, welcome to Clinical Microbiology for the Infection Preventionist. I'm Tommy Babadi and I'll be your instructor for this course. So here's a quiz. There's a hunter that came to your hospital a few days ago after going hunting in the summer. He has an ulcer on his hand and an enlarged lymph nodes in his axilla, plus he's got bilateral conjunctivitis. The doctor suspects tularemia and orders for a culture. What is your infection control concern in this situation and what would you recommend? So let's talk about Robert Koch, who is a German physician and a prominent bacteriologist that lived around the 1800s. He did most of his work on tuberculosis and anthrax. So Dr. Cox came up with a postulate from all his work that should be satisfied for us to say that an organism caused a particular disease. And his postulates has four different parts. The first and most important one is that an organism must always be present in every case of that disease. So since we say tuberculosis disease or infection is caused by a bacteria called Mycobacterium, it says for every case of tuberculosis, we must be able to find Mycobacterium present. So that is something that must be satisfied all the time for us to say an organism caused the disease. Then the next thing is we must be able to isolate that organism from the person who has the disease, and we must be able to grow that organism in a pure culture. So that is where the principles of culturing and culturing came from. We must be able to culture it. And the third principle is that when we collect samples from the organism that we've cultured and we inject it in the lab in a healthy, susceptible person, says that person should also come down with the disease. And then fourthly, when the second case comes down with the disease, we should also be able to culture it again in that person and be able to identify the same mycobacterium in the second case. So if these four postulates are satisfied, then we can say without a doubt that this organism is definitely what is responsible for this disease. So that is Koch's postulate, and it's something to always remember at the back of our hands as we go through this course for the rest of the semester. Then Rothman has another idea to broaden Koch's postulate about the theory of causation. So let's say we want to know what caused stomach cancer. He represents the concepts of causation as different pieces of pizza. So for stomach cancer to occur, a piece of pizza must be complete. And once this happens, stomach cancer is going to occur. And in his own piece of pizza model, there are different slices that make up this pizza. Each of these slices of pizza don't have to all be present at the same time. They could be present at different points in time, but once all the pieces of the slice of the pizza come together, no matter how they occurred in time, once it's complete, it says all the necessary and all the necessary components have been met, and now we have a sufficient cause for stomach cancer to occur. So, take for example this first scenario of pizza that can cause stomach cancer. You need H. pylori to be present at some point. You need the patient or the person to have diet rich in nitrates as their diet. And then you need them to have a high salt intake, plus an always present component, which is X or the unknown factor. So in this particular model, once all these three plus unknown factors of the slice of pizza have occurred, irrespective of the time frame in which they occur, either sequentially or one after the other, once this is complete, then it says we have our sufficient complete causal model. That means stomach cancer is definitely going to happen. Now, these slices of pizzas that make up a complete pizza can vary from case to case. In the second case here, instead of having 4 slices, we have 5 slices for it to be complete piece of pizza. We have H. pylori, we have the unknown, and this one introduces smoking into the picture and vitamin C deficiency. And once we have all 5 components complete, irrespective of the sequence, then a complete or sufficient pizza gives rise to an inevitable stomach cancer in this scenario. And then we have a fourth one as well, which includes smoking and high salt intake. Now, when we take a look at all these 3 different complete or sufficient pizza pies for stomach cancer to occur, we have one that is a constant. H. pylori is here, H. pylori is here, and H. pylori is here. So when these kinds of situations occur, which most of the time in the theory of causation, it happens in infectious diseases about 90% of the time, there has to be a necessary course. That means in this theory of stomach cancer causation, if there's no H. pylori, the piece of pizza would never be complete. So for stomach cancer, we say H. pylori is necessary. So necessary courses is almost exclusive to infectious diseases. In most other theory of causation, we don't have just one particular single entity that is a necessary course. Say, for example, in lung cancer, smoking has a very, very high risk for developing into lung cancer in those that smoke. But smoking is not necessary for you to have lung cancer. Someone could have lung cancer from, say, work exposure to asbestos, even if you've never smoked. So asbestosis is an alternate component cause that, with a bunch of other things, can make a sufficient causal pie to lead to lung cancer. So that's just an example to say that infectious diseases, it's almost exclusively the part of causation that has a necessary course, without which the events will never be complete and the outcome will not take place. And here's another example to show proximate, intermediate, and distal causes. Let's say we want to know what caused stroke. Now, the proximate cause of stroke in this particular individual is hypertension, plus underlying genetic susceptibility, which we cannot change. But let's say we're interested in the hypertensive part of it. So hypertension plus a genetically susceptible individual leads to stroke. But then we want to know what caused hypertension, even in a genetically susceptible person. Then we have the intermediate causes. So the person is obese, the person is taking high salt intake, under a lot of stress, and living a sedentary lifestyle, plus some unknown factors, as we saw before. So all of this makes this complete pizza pie, and once this is sufficient, it leads to hypertension, which leads to stroke. So this represents an intermediate course for stroke. And let's say we want to know what made this individual obese and living under stress and having a high salt intake and living a sedentary life. Then there's another set of courses, which is low social class. So low social class leads to obesity because you don't have enough money to eat as healthy as you would like, and you don't have enough money to live in an environment where you have good access to gym and where your housing is made up of good networks of roads, there's good security where you can easily exercise. So low social class leads you to completing the intermediate course, which leads to the proximate course for stroke. Essentially, a low social class is a distal cause for stroke. And there are 9 guidelines that are important for us to say in the theory of investigation that X leads to Y as a causative agent. The first and the most important is the temporal relationship. That means X must always come before Y if we're going to say X caused Y. So you must always be exposed to mycobacterium before you can ever get TB. If you don't get exposed to the bacteria first, we can never say you've had a TB. So that is the most important thing. You must always get the offending agents coming before the disease happening. The next thing is the strength of the association. If those who smoke are 10 times likely to have lung cancer compared to those who smoke, then 10 times risk is something for us to think about because that is a lot. So the strength and dose response relationship. If the more you smoke, the more likely you're going to have, let's say, lung cancer, for example, then that is also an indication that maybe there is a causal relationship there. And replication of the findings means it's not just particular to you. If we do the same thing somewhere else, we should get the same results. And biological plausibility just means that it has to make sense biologically. Just like somebody cannot come tomorrow and say drinking too much water causes TB. That is not biologically plausible. And the rest of the theories for the guidelines for causation are not so critical to infectious diseases, but I still think for your epidemiology classes, you should just remember them. So let's just talk about and go over the basic bacteriology, which we all most likely know already, but we're just going to have this as a refresher. So in the structure of the bacteria, there's the peptidoglycan, which gives the bacteria rigid support and protects it against osmotic pressure, and this prevents it from getting destroyed by osmosis. This peptidoglycan has a transpeptidase cross-linkage and a sugar backbone, which gives it its strength. Then we have the cell wall. The cell wall is present in gram-positive bacteria and which serves as its major surface antigen. And the chemical composition of the cell wall also is the lipoteichoic acid, which induces tumor necrotic factor and interleukin-1. It also serves as a support for the peptidoglycan. Then for the gram-negatives, they have an outer membrane. This outer membrane serves as a site for endotoxins, which contain the lipopolysaccharide LPS and serves as its major antigen. And it also induces tumor necrotic factor and interleukin-1. Then we have the plasma membrane, which serves as transport of enzymes. We have the ribosomes, which helps in protein synthesis. We have the capsules in some bacteria that help it to evade the host defense mechanism. We have the pilus and the fimbriae, which can serve as a tool that the bacteria uses in adhering to the presence of foreign bodies in the host. Say, for example, Foley's and central lines. So they can use this fimbriae to attach and adhere strongly to this foreign body in our system. Then the flagellum is used in motility. Then the spores are used to survive harsh conditions, to be resistant to dehydration, heat, and chemicals. The plasmid contains various genes that can be used for antibiotic resistance. And the glycocalyx helps it to also mediate adherence to surfaces, especially foreign surfaces like Foley's and central lines as well. Just reviewing the basic taxonomy. When we say bacteria is coccus, it just means it's circular, it's round. Famous ones are Staphylococcus, Streptococcus for gram-positive, and Neisseria for gram-negative. Bacillus just means rods. And examples are Clostridium, Mycobacterium, Gardnerella, Listeria, and Bacillus, which are the gram-positive rods. Then the gram-negative rods, which are always very troublesome rods, have different classes, which I'll just leave you to study those. Enterics are E. coli, Shigella, Klebsiella, and Pseudomonas. Those are the important ones, plus Bacteroides. The branching filaments, which are the nocardia, which is weakly acid-fast, and the actinomyces. The spiral bacteria's examples are spirochetes. The common spirochetes are Borrelia, which causes Lyme's disease, and Treponema, which causes syphilis. And some that do not really have a cell wall, which is Mycoplasma. The Mycobacterium, which causes TB, is said to have a high lipid contents and contains mycolic acid, which gives it this strong resistance to antibiotics in general and gives it a special characteristics. Here's what a general picture of bacterial cell structure looks like. We have the cell wall in the gram-positive, and we can see the cell wall in this gram-positive is a huge layer of cell wall compared to the very thin cell layer of cell wall in the gram-negative. This is what stains purple on the gram-positive bacteria, and this is what stains pink in the gram-negative bacteria. Now, the lab's first step in trying to identify a bacteria is to do a microscopy. A microscopy is a quick way to assess and view the structure, which gives us the shape of the bacteria, and the physiology, which essentially for microscopy is the staining pattern of the cell. Now, only the genus can be identified with the first step of microscopy. We cannot identify up to the level of the species. We have to do a culture to do that. And staining techniques are used to be able to get meaningful bacterial architecture on microscopy. The most popular staining technique is the gram stain. It often takes about 3 minutes. The first step in a gram stain is to use a crystal violet, which is a purple dye. After you do a heat fixation of your specimen on the slides, then you add the crystal violet. After the crystal violet, you add an extra chemical, which is the iodine, which is like a mordant, to make the crystal violet stay and adhere to the cells. By this time, most of the cells are purple because they've absorbed the crystal violet. Then the next stage is you want to decolorize with the help of a decolorizer that acts like a bleach. You want to decolorize away the crystal violet to see if it takes away the color. And then finally, you add a pink stain, which is the safranin stain. And that completes your gram stain in about 3 minutes. So if it is a gram-positive bacteria, it should not get decolorized. The purple should remain. And if it's a gram-negative, it will get decolorized because it has a thin cell wall and it's going to hold on to the pink stain. Here's the algorithm of gram staining for gram-positive. So if we look under the microscope while doing our gram stain, if we see pink bacteria, then we know that that is a gram-negative. If we're seeing purple, we know that that is gram-positive. Then we look under the microscope to see the morphology. If they're cocci or circular, if they're rods or bacilli, or if they're branching filaments. So if they're branching filaments, we want to know, we'll do an extra stain to say, are they acid fast? If they're acid fast, then we know that that's nocardia. If they're aerobes but they're not acid fast, we know that that's actinomyces. But if they're cocci, then the next thing we want to add is, that's if they're cocci, then that ends our microscopy, then we want to proceed to culture. At this point, the microbiologist can call the doctor and say, hey, I see a gram-positive cocci, and then that should give the doctor an idea of the likely organisms they could be dealing with. So the lab goes on to do a culture, and after a few days, say 48 hours of culture when they have preliminary growth, then you do a catalyst test. So if it's catalyst positive, it will come most likely in clusters and you see that this is Staphylococcus aureus, or just staph in general. Then you do an extra step. You add coagulase tests. And if it's coagulase positive, then you can zero down to say this is Staphylococcus aureus. If it's coagulase negative and catalase positive, then we know that it's likely to be the other two staphs, which is Staph epidermidis or Staph saprophyticus. To differentiate between epidermidis and saprophyticus, you add novobiocin. If it's sensitive to novobiocin, which is novobiocin kills the organism, then it's Staph epidermidis. If, if novobiocin doesn't kill the organism that is resistant, then you know it's saprophyticus. If while doing the catalyst test it becomes catalyst negative, then you know that, oh, this is a gram-positive cocci and it's streptococcus. Then to differentiate what kind of streptococcus it is, we rely on the hemolysis pattern on the culture. There are three different hemolysis patterns that can be seen. Alpha hemolysis, which is partial and appears slightly greenish. Beta hemolysis, which is completely hemolyzed and it appears clear. Or no hemolysis, which is the gamma hemolysis. So say on the slide or on the culture plates, we see partial hemolysis, so there's a greenish tinge. We say that's alpha hemolysis. So to narrow down on the species of the streptococcus with the alpha hemolytic characteristic, we had optochin, which is like an antibiotic. So if optochin works and kills the bacteria, then we know that is strep pneumonia. If optochin doesn't work and is resistant to optochin, then we know that is viridans streptococcus, which is strep mutans. So let's say there's complete hemolysis. That's a beta hemolytic streptococcus. We want to know, is it a group A streptococcus or a group B streptococcus? So let's say it's a gas, group A streptococcus. We want to know what kind of streptococcus it is. We add bacitracin, which is also like an antibiotic to it. So if bacitracin kills the streptococcus, then we know that that is a group A streptococcus. If it's resistant, then we know that that is a group B streptococcus, which is Strep A galacea. So let's say on hemolysis there is zero hemolysis, which is gamma. Then we want to know, is it group D streptococcus or is it Strep bovis? To do that, you culture it in bile and 6.5% normal saline. If it grows on 6.5% normal saline, then we know that it is E. faecalis. If there is no growth on 6.5% normal saline, then we know that that is Strep bovis. For the gram-positive rods, we have a few examples here. We have Clostridium, which is an anaerobe. We have Bacillus, which is an aerobe. And we have the Mycobacterium. So the Mycobacterium is an acid-fast bacilli. So after looking under the rods, and we see that they publish. So if we suspect Mycobacterium, we do an extra stain, which is the acid-fast stain. So here is a walkthrough of the gram-negative algorithm. Again, if we do a gram stain and we see purple, we know it's gram-positive and we go the previous way. So, but if it's pink, there are different ways this could go. So if it's pink and then we do an oxidase test and it's oxidase positive and comma shaped, then we want to do your urase test to see if it's urase positive, then it's H. pylori. If it grows in alkali, then that's cholera. And if it grows in 42 degrees Celsius, that's Campylobacter. If we look under the microscope with a gram-negative bacteria and we see diplococci, which is just two circles, they're just two circles in pairs of twos, that's Diplococcus. Then we want to differentiate what kind of Diplococcus is this, and we use a Malthus test. If it ferments on maltose, then that's Neisseria meningitidis. It turns the culture pink. If it doesn't turn it pink, then that's Neisseria gonorrhea. If under the microscope they're coccoid rods, that means they look like they're circular, they also look like they're rods. They look circular, they look like they're rods. Then that is Haemophilus influenza or Bordetella pertussis. If they are gram-negative rods, which are a very important group of organisms in infection prevention, then the next step we want to take is we want to do a lactose test to differentiate if it is lactose fermenting or non-lactose fermenting. If it is lactose fermenting, then it could be either fast or slow. Fast fermenters are Klebsiella, E. coli, and Enterobacter. Slow fermenters are Citrobacter, Serratia, and we have a few others that are not so critical. That's why I didn't list them there. So lactose fermenters essentially would turn the agar pink into wine, which is a sign of fermentation. So if it is a non-lactose fermenter, the next steps you want to do is an oxidase test. If it's oxidase positive, non-lactose fermenting rod, gram negative, then you know that that is Pseudomonas. If it's oxidase negative, then you know that that is either Shigella, Salmonella, or Proteus, and you take an extra step in differentiating which is which. Let's just quickly talk about culture. Now, the purpose of culture is to identify the bacteria up to the level of the species, which microscopy will not tell us. If there's a growth of more than two organisms, this often means contamination, and we say this is polymicrobial. Say you do a culture and you see E. coli, Pseudomonas, Streptococcus, and you see Staph aureus, that is most likely contamination. Excuse me. Now we can quantify growth based on how many colonies are counted from the culture. And we use colony forming units. It could be 10,000, 20,000, 50,000, 100,000, or greater than 100,000. And this is important depending on what kind of specimen is being collected. If it's urine, for example, and you have greater than 100,000, that becomes important and could be a greater indication that this is indeed an infection. But if it's in the blood, just a few thousand colonies could as well be clinically significant. So cultures can also be quantified based on how much colony forming units you have, and that gives a sense of how much infection is going on. Now there are different growth medias. There's the general or the basic growth media, which tends to grow most organisms. Examples of those are blood agar, chocolate agar, or brain heart infusion agar. And there are some others which are selective media. We have Maconche, LJ medium, Hyoscine methylene blue, and mannitol salt agar. These are selective media and they don't necessarily grow every single type of organism. And so here is an example of a gram-positive and gram-negative lactose fermenting and non-lactose fermenting. As you can see here, on the left side is lactose fermenting, which is pink or wine. And on the right here, you can see it's just colorless. So based on this, we can have an idea what kind of organism this is. Pink means lactose fermenting. And examples here, you can see this is E. coli, which is gram-negative rod, and it's lactose fermenting. Another example here is Enterobacter, and another example not shown here will be Klebsiella. You can see these ones that are non-lactose fermenting, which is Proteus and Salmonella and Staph aureus. So tuberculosis is grown on a special media called the low-extingency medium for Mycobacterium tuberculosis. And this medium is made up of beaten eggs plus mineral salt and a special greenish stain which is called the malachite green. And you can see that this is the top of this culture is made in a slanted way so that it can give a higher surface area to allow bacterial growth. And on this high surface area, we can see an actual growth of Mycobacterium tuberculosis. That was just a quick overview, and I hope that this has helped to serve as a refresher for your memory in the basic science of bacteriology. I hope that as you get to read your textbooks, a lot of the things you probably know before will be coming back to you, like riding a bike. And I look forward to hearing your discussions on the discussion board. So here is a quick assignment, and I'd like to hear your thoughts on this. It says, what organisms are highly infectious, stroke hazardous for lab personnel to culture? So I'd like to hear your response in the discussion board. And I hope you have a good rest of your day.